I want you to imagine your kitchen counter right now. On it, you've accidentally left a carton of milk out overnight.
Yeah, happens to the best of us.
Right, exactly.
Yeah.
And right next to it is a fresh artisanal bowl of yogurt. Now, instinct tells you to grab that milk and pour it straight down the drain because, it's spoiled.
Oh, absolutely.
But you'd happily grab a spoon and eat the yogurt. And what is truly wild here is that the exact same microscopic organisms, like the lactic acid-producing bacteria, could be at work in both of those containers.
That's the crazy part. Biologically speaking, the milk and the yogurt are undergoing incredibly similar processes.
I mean, the only actual difference between what we call spoilage and what we call fermentation is human intent.
Yeah, the microbe certainly doesn't know it's making a highly sought-after gourmet product for you.
Right, it doesn't care.
No, nor does it know it's ruining your breakfast.
Yeah.
It is simply metabolizing sugars in its environment to survive. And, that exact dynamic is the foundation of our mission in this deep dive. We are exploring the intertwined worlds of food microbiology and industrial microbiology, which is really the study of this invisible, silent workforce all around us.
It's everywhere.
It really is. It's a workforce that spoils our food, ferments our favorite snacks, and manufactures our life-saving medicines at a staggering industrial scale.
It really is an invisible turf war happening on every surface, in every pantry. And the way we think about this turf war has completely changed, right? Because if you looked at a textbook from, say, the mid-20th century, microbes were basically cast in a morality play.
Yes, exactly.
You had the good guys, like starter cultures and cheese, and you had the bad guys, like the spoilage organisms and pathogens. It was a very black and white hero and villain narrative.
And that old paradigm has shifted entirely, particularly since the 2000s. The real catalyst for this shift was the advent of metagenomic sequencing.
Wait, let me stop you right there. Metagenomic sequencing sounds incredibly dense. Can we get an analogy for what that actually looks like in practice?
Sure, yeah. Think of it this way. In the past, studying microbes was like trying to understand the ocean by catching one fish at a time, bringing it to a lab, and looking at it in a tank.
Okay, pretty limited.
Very. But metagenomic sequencing is like scooping up a bucket of ocean water and instantly reading the DNA of every single organism in that bucket all at once.
Oh, wow.
Yeah. So when scientists gained the ability to do that, the picture became vastly more complex. I mean, if you sequence a cheese rind or a jar of fermenting kimchi today, you don't just find one or two quote-unquote good species working in isolation.
You find a whole city.
Exactly. You find a highly complex, constantly shifting microbial ecosystem. You might have dozens or even hundreds of species interacting, competing, and collaborating. They just don't map onto our human concepts of good and evil.
Right.
We now understand these environments as ecosystems under intense selective pressure.
Okay, let's unpack this. Because to understand this modern ecosystem view, we actually have to go back to the 19th century, to the exact moment humans realized they were even in a microscopic competition for their food.
Yeah, to understand how we attempt to control these ecosystems today, we really have to look to Louis Pasteur in the 1850s and 1860s.
The milk guy.
Well, yes, but at the time, it was actually the French wine and beer industries that were in crisis. They were losing massive amounts of money because vast batches of their product were unpredictably turning sour or bitter or ropey.
Ropey wine sounds absolutely disgusting.
It was not great. And the prevailing scientific consensus of the day was the theory of spontaneous generation.
Oh, right.
This is the belief that life could just, spontaneously arise from non-living organic matter, and that chemical breakdown was just a natural, non-biological event.
Which seems ridiculous to us now. But at the time, if your wine's spoiled, you just thought the wine was chemically destined to do that.
Exactly. And Pasteur completely dismantled that idea. He was commissioned by industrialists and later the French government to figure out what was actually going wrong.
And he did this through those famous swan neck flask experiments, right? Between like 1859 and 1864.
That's right. Pasteur boiled broth in a flask with a long curved neck shaped like an S.
Okay.
The curve allowed air to enter, but trapped any dust particles or microbes in the dip of the glass. Because the dust couldn't reach the broth never spoiled.
Just because of the shape of the glass. That is so elegant.
It really is. And this proved that both fermentation and spoilage were caused by living microorganisms falling in from the air or resting on equipment, not spontaneous chemistry.
Right.
This single discovery established the first principle of food microbiology, which is that microbes are the cause of spoilage. And out of this industrial problem-solving came pasteurization.
Which is just the process of applying brief heat to kill those spoilage organisms without completely ruining the flavor profile of the liquid, right?
That is the mechanism, yes. It was patented in 1865 for wine, and obviously later became the standard for milk. Pasteury proved that microbes need the exact same basic things to survive that you and I do.
Like what?
Well, they need water, they need nutrients, they need the right temperature, and they need the right environmental conditions, like specific pH levels or oxygen levels.
So preserving food isn't some complex modern chemical magic. It's basically medieval siege warfare.
I like that.
I mean, we are just systematically cutting off their supply lines. Like, if they need water, we salt the meat or dry the fruit, which physically pulls the available water out.
The siege warfare analogy is highly accurate. Microbes are simply competing for the very same food resources that we want to eat.
Right.
Food preservation techniques are just systematic methods of denying them those essential requirements.
But hold on, if we are talking about medieval sieges, let's talk about the refrigerator.
Okay.
If I put leftover chicken in the fridge or the freezer, it lasts way longer. It definitely feels like I'm dropping a winter siege on them and killing them off.
That is one of the most persistent misconceptions out there. Dropping a temperature does not kill most harmful bacteria.
Wait, really? It doesn't.
No, it simply slows down their enzyme kinetics.
Enzyme kinetics? You mean like the microscopic gears of the bacteria?
Think of enzymes as the physical machinery that allows a bacterium to eat, grow, and divide. When the temperature drops, the chemical reactions powering that machinery grind to a near halt. You're pausing the siege, not winning it.
Oh, wow.
The pathogens just go dormant. As soon as the food warms back up, the gears start turning again. And worse, there are exceptions that don't even go dormant.
Wait, what exception? I thought freezing temperatures were a universal pause button for biology.
Not for a class of bacteria known as psychrotrophs. These are organisms that have evolved to thrive in cold environments.
Oh, no.
Yeah, Listeria monocytogenes is a prime example. It can and will happily, albeit slowly, multiply at the standard 4 degrees Celsius, which is 39 degrees Fahrenheit, of a normal home refrigerator.
That is terrifying.
It is. This is why Listeria remains such a major, persistent concern in ready-to-eat foods like deli meats and soft cheeses that are stored cold for extended periods.
Wow.
You put it in the fridge thinking you've paused the invasion, but the Listeria is slowly building its army right there in the crisper drawer.
So if the whole strategy is to just trap them in a harsh environment, what happens if we mess up the math? Like, is it possible to accidentally build a luxury resort for the absolute worst microbes?
You've hit on the central paradox of preservation. And for that, we have to look at Nicholas Appert, who invented food canning back in 1809.
Okay, early 1800s.
Yeah, he developed the process to feed Napoleon's armies, basically sealing food in glass jars and heating it. What's amazing is that he did this decades before Pasteur came along.
Right, so he was just guessing.
Completely. Appert had no idea why it worked. He didn't know about microbes. He just knew that heat and a seal kept the food from visibly rotting.
But by boiling that jar and sealing it, he accidentally created the absolute dream home for a specific nightmare of a microbe, right? Clostridium botulinum.
Clostridium botulinum is an anaerobic, spore-forming bacteria. Anaerobic means it can only thrive in the absence of oxygen.
Which a sealed jar definitely is.
Exactly. When you seal a jar, you strip away the oxygen. If the food inside is also low in acid, like green beans or soups, you have created a flawless incubator. But what makes it truly formidable is that it forms spores.
Like microscopic armor plating.
A biological bunker is a good way to look at it. When the bacteria senses stress, like the heat from boiling water, it bundles its DNA into a hard, nearly indestructible shell.
Wow.
Once the heat stops and it finds itself in a nice, oxygen-free, low-acid jar, it hatches from that spore. And as it grows, it produces one of the most potent neurotoxins known to humanity. To put it in perspective, just a few hundred nanograms of botulinum toxin can be lethal to an adult human.
A few hundred nanograms. That is almost unimaginably small. And the stakes of getting this wrong were highlighted tragically in 1971 with the Bon Vivant Vichyssoise soup case. It was a commercial canned soup company. The soup was undercooked during the canning process, meaning the heat wasn't high enough to crack those armored spores.
Right.
A man in New York died, another person was paralyzed, and the public fallout was so intense that the entire company collapsed within a matter of weeks.
It demonstrates that you cannot rely on visual cues. The soup looked fine, smelled fine, and the can was sealed.
Okay, here's where it gets really interesting. Especially for you listening, if you or someone you know is into home canning, there is a massive, incredibly dangerous misconception that if the lid of a canning jar pops and forms a vacuum seal, the food inside is completely safe. I
Yes.
hear this all the time. But if it pops, there's a vacuum, meaning nothing is getting in or out, right? Doesn't that mean it's safe?
Not at all. A vacuum seal only tells you that the jar is airtight because the cooling air inside contracted. It
Yep.
tells you absolutely nothing about whether the contents reach the necessary temperature to destroy those botulinum spores.
Oh, I see.
If you are canning low-acid vegetables in a standard boiling water bath on your stove, the water only reaches 212 degrees Fahrenheit. You get a perfect vacuum seal, but those spores can easily survive that temperature.
That is so
You
scary.
just wake up in that perfectly sealed, oxygen-free jar and start pumping out toxin.
So how do you actually kill them if boiling water isn't enough?
True safety in canning comes from precise time temperature pressure standards. In the 1920s, the National Canners Association developed what is known as the botulinum cook.
The botulinum cook.
Yes, and to destroy those spores, you have to use a pressure canner. By trapping the steam in a pressurized vessel, the physics of the water change, allowing the liquid to reach temperatures well above the normal boiling point, typically around 240 degrees Fahrenheit.
Okay, so it's a structural thing.
It is a mathematical engineering standard to break the spores, not just a matter of boiling a jar until it seals.
So we've talked about how we wage war against microbes by cutting off their supplies or crushing them under pressure. But what if, instead of fighting them, we recruit them?
I like where you're going with this.
I mean, if we can't kill all the microbes in the turf war, why not just rig the game and pick the winner?
That is the core mechanism of fermentation. It is applied competitive exclusion. When you make sauerkraut by salting cabbage, or you make yogurt by adding a starter culture to milk, you aren't trying to create a sterile environment.
Right.
You're deliberately giving a specific group of microbes, the lactic acid bacteria, or L. labile, a massive head start. Organisms like lactobacillus.
Because they're incredibly fast eaters, right? It's like a scorched earth policy. They consume the resources so nothing else can.
They rapidly consume the available sugars and convert them into lactic acid. By doing this, they drop the pH of the food down to around 3.5 to 4.5.
And acid is bad news for the other guys.
Mechanistically, acid denatures proteins. It unravels the cellular machinery of most spoilage organisms and deadly pathogens. They simply cannot survive in an environment that acidic. The lactic acid bacteria win the resource race, and in the process, they literally poison the environment for all their competitors.
But how on earth do you take a countertop process, like, a jar of kimchi, and scale it up to supply an entire continent? It seems like an impossible leap.
The leap was actually forced by the desperation of global conflict. Industrial microbiology emerged as a vital engineering discipline during the World Wars, specifically in reaction to severe material shortages.
Makes sense.
During World War I, Britain desperately needed acetone to manufacture cordite, which is a smokeless propellant for artillery explosives. In 1916, a chemist named Chaim Weissman developed a process using the bacteria Clostridium acetobutylicum to ferment massive quantities of acetone.
Wow.
It was the first true industrial-scale bacterial fermentation.
And then World War II pushed the field even further with the race for penicillin, right? Alexander Fleming had discovered penicillin by accident back in 1928, but he couldn't figure out how to mass produce it from his original finicky mold.
Exactly, he was stuck.
By 1943, the Allies needed antibiotics for the front lines, which triggered a massive government-funded crash program at the Northern Regional Research Laboratory in Peoria, Illinois. This is such an incredible story. They were literally screening moldy produce sent in from all over the world, desperately looking for a better strain.
It was a massive dragnet.
But the breakthrough didn't come from an exotic jungle. It came from a moldy cantaloupe found at a local fruit market right there in Peoria.
I love that detail.
Right. They isolated a superior penicillium strain from that melon, mutated it, selected the best survivors, and that specific mold became the ancestor of nearly all industrial penicillin used in the world since.
What's fascinating here is how industrial microbiology relies on finding the right natural ally and optimizing its native behaviors. And this isn't just about wartime antibiotics. The same logic applies to the food in your pantry right now.
Like what?
Think about citric acid, the ingredient that gives soft drinks and candies their tart flavor. Originally, we extracted citric acid from actual lemons grown in Sicily, which was incredibly slow and expensive.
But then, in 1917, a food chemist named James Curry made a wild discovery. He found that a common mold, Aspergillus niger, could be coaxed into excreting massive amounts of citric acid.
And you have to look at why the mold does this. It isn't trying to make our soda taste better.
No, I imagine not.
It secretes citric acid as a metabolic weapon to bind up trace minerals in its environment, hoarding nutrients so competing microbes can't grow. Pfizer commercialized this process in 1919 using giant submerged fermentation tanks, giving the mold the perfect mix of sugars to trick it into overproducing this acid.
Today, more than half of the entire global supply of citric acid comes from this industrial mold fermentation process. So when you drink a citrus soda, you are largely tasting a fungal byproduct, not an extract of fruit.
Yep.
And it goes beyond acids. Monosodium glutamate, or MSG, is often assumed to be a purely synthetic chemical concocted in a lab. In reality, modern industrial MSG is produced via bacterial fermentation, primarily using a bacterium called Corynebacterium glutamicum. It is mechanistically much closer to brewing beer or making yogurt than it is to artificial chemical synthesis.
Which is something most people don't realize.
So what does this all mean? We are talking about taking a microscopic organism from a tiny petri dish and scaling it up to a 100,000-liter bioreactor. You're dealing with oxygen transfer, heat removal, preventing outside contamination. It's not just biology anymore, it is hardcore chemical engineering.
Yes, it is.
And when you are producing things on that scale, the risks of a microscopic mistake scale up with it.
And this raises an important question about how we ensure safety at such massive scales. For most of the 20th century, food safety was entirely reactive. Regulators simply inspected the finished product at the end of the line.
Just hoping it was safe.
Right. But in the 1960s, a paradigm shift occurred, driven by the space race. The Pillsbury Company, working alongside NASA and the U.S. Army, had to figure out how to make food perfectly safe for astronauts.
I can imagine. A single case of foodborne illness in a tiny zero-gravity space capsule would be an absolute nightmare. But you can't just cross your fingers and hope the final freeze-dried meal is safe.
You cannot. And you can't test every single meal, because then there would be nothing left to eat. So they developed HACCP hazard analysis and critical control points.
Okay, HACCP.
Instead of just testing the final product, HACCP requires engineers to map out the entire assembly line, identify every single weak point where contamination could enter, and impose strict mechanical or chemical controls at those specific points. It shifted the entire global food safety system from end-of-line inspection to source prevention.
But even with systems like HACCP, there is constant tension between traditional natural production and these strict safety controls. You see this intensely in the debate over raw milk cheese. We have to look at this impartially because both sides have deeply held arguments. On one hand, traditional cheesemakers argue that using unpasteurized raw milk preserves a complex, terroir-driven microbial community.
And terroir, in this context, refers to the unique flavor profile imparted by the specific local environment, so the soil, the climate, and the native bacteria of the region. They argue pasteurization destroys that nuance.
Right.
On the other side, food safety regulators point to the raw data, noting that unpasteurized cheeses have repeatedly caused deadly outbreaks.
And regulators are heavily influenced by historical tragedies.
They are. In 1985, there was a devastating outbreak linked to Jalisco brand soft cheese in Southern California, which was made with unpasteurized milk.
Oh, that was a big one.
It was. The cheese was contaminated with Listeria monocytogenes. It caused roughly 142 illnesses and resulted in dozens of deaths, including miscarriages and stillbirth.
It's tragic.
It is. That single event is the direct reason why the U.S. currently mandates a 60-day aging rule for any cheese made from unpasteurized milk. The underlying logic is that the hostile, acidic environment of the aging cheese will eventually starve and kill off any pathogens over those 2 months.
And it's crucial for you to understand that terms like natural or organic do not give you a free pass from microbial risk. There is a huge myth that organic food has no toxin risk. But naturally occurring toxins, like aflatoxin, which is produced by a mold that grows on stored grain and peanuts, will form on organic crops just as easily as conventional ones.
Absolutely.
Biology doesn't care about our marketing labels.
Biology is also entirely unforgiving of human negligence. When the engineering fails, or when standard practices are ignored, the microbes exploit the vulnerability immediately.
Always.
The 2008-2009 Peanut Corporation of America outbreak is a stark example. Internal records revealed that executives knowingly shipped peanut products that had already tested positive for salmonella.
Which is just wild to me.
It is. That decision contributed to at least 9 deaths and over 700 illnesses. The company's owner was ultimately sentenced to 28 years in prison.
which is one of the harshest sentences ever handed down in a food safety case. But sometimes it isn't intentional, it's just bad engineering. The 2011 Jensen Farms cantaloupe outbreak is a perfect example of this. 33 people died from listeria.
Yeah.
The root cause wasn't the farm fields themselves. It was traced back to poorly designed post-harvest washing equipment that couldn't be properly sanitized. Water pooled in the machinery, bacteria grew in the stagnant water, and the machine itself literally coated the cantaloupes in listeria as they rolled through.
A perfect storm.
It proves that keeping food safe is just as much an engineering discipline as it is a biological one.
If we connect this to the bigger picture, the stakes of food and industrial microbiology become incredibly clear. The World Health Organization estimates that hundreds of millions of people fall ill from contaminated food globally every single year, with hundreds of thousands of deaths.
That is staggering.
This isn't just an academic curiosity. It is a frontline global public health issue on par with major infectious diseases.
But on the flip side, that exact same invisible workforce is the engine of the modern world. Microbes are fermenting the statins that lower your cholesterol. They are producing the enzymes in your laundry detergent that break down stains. They are manufacturing the recombinant insulin used by diabetics.
It touches everything.
Even mRNA vaccines and precision fermented dairy that doesn't require cows. All of it relies on the industrial fermentation infrastructure that this field pioneered over the last century. I mean, human progress is completely tied to how well we negotiate with these microscopic ecosystems.
It is a profound realization. Which brings me to a final thought I want to leave you with, one that directly impacts your daily life. Earlier, I mentioned Ilya Mechnikov, a Nobel laureate who, in 1907, theorized that Bulgarian peasants lived so long because they ate yogurt rich in lactobacillus.
Right?
That single idea planted the seed for today's massive, multi-billion-dollar probiotic industry.
So the next time you are standing in the supplement aisle, staring at a highly priced bottle of probiotic capsules, promising to balance your gut, think about this. There is a major live scientific debate happening right now about whether those ingested bacterial strains actually colonize a healthy human gut or if they just pass right through.
Exactly.
Rigorous strain tracking studies are showing that if you already have a stable gut microbiome, durable colonization by a new store-bought probiotic strain is the exception, not the guaranteed rule. It raises a fascinating question. Are you buying a permanent microbial ally to join your internal ecosystem, or are you just paying for some very expensive, very temporary microscopic visitors?
It requires us to look past the marketing and understand the actual ecology happening inside us.
Thank you for joining us on this deep dive. We hope it makes you look at your kitchen, your pantry, and your medicine cabinet in a completely different light. Keep questioning the invisible world around you, and maybe go put that milk back in the fridge.